Thank you. Okay, so good morning, everyone. Very delighted to be with you today at the ABIC community meeting. Today, I will share with you the Algerian model for collaborative infrastructures. So first, I will talk to you about the national framework for shared technological. Okay. Okay. So we have a national framework for shared technological platforms, and this includes technological platforms, technical facilities, incubators, etc. So the aim of this is to pool all specialized equipment and expertise and make it accessible to all. It has an online portal, so you can access the online portal and choose which facility, based on which technique you need to use, or the city or the service that you need to access. Okay, for all these this framework, the management is done by a platform manager, as long as a technical and scientific council that also includes socio-economic partners. And then as for the access, there are two frameworks. So either free for service or fee for service. So for the free of service option, it's based on collaboration. So most of our users come through collaborations, and they get access to all the infrastructures free of charge. As for the users' training and maintenance, well, they are usually done regularly by workshops and also by collaborating with the manufacturers. So anytime we purchase an equipment, we always have the option to collaborate with the manufacturers on long term. We also have a very strong biosafety and biosecurity team, a certified team by Sandia Labs. We also had an IFBA Heroes Award, and we also applied to host a regional center of excellence in biosafety and biosecurity for North Africa region. Okay, so now I'm going to talk to you about the bioengineering platform. So our bioimaging is a unit in the bioengineering platform. So the bioengineering platform is a platform specialized in nano and microfabrication, the development of lab-on-chip devices. How it came about? Well, it all started with a project of Professor Ammar Azioun. Professor Ammar Azioun is a pioneer in lab-on-chip technologies. He spent 25 years in Europe and created three startups pioneering these technologies. And then he came back with all this knowledge to Algeria and created this platform. So you can see here that it took it's going to take almost 10 years for the platform to be fully operational. So right now, we have all the equipments, some of them or most of them installed, but on a different facility. So the only step that still remains for the platform to be fully operational is to be done with the clean rooms set in, and this will be done by the beginning of 2026. Otherwise, right now we are putting all the equipment in clean rooms that are from another platform at the Biotechnology Research Center, which is the genomics platform. Okay, so as I said, our bioimaging is a unit and not a facility. So the facility is bioengineering, and then we have the bioimaging unit inside it. And to give you a glimpse of what type of equipment we have at the bioimaging unit. So we have a cytonaut, different cytonaut lenses-free systems that go into the incubators, and they are specifically used to monitor cells or spheroids label-free. So to get live data inside the incubators. And then we also have a TÜV super resolution microscope, motorized microscope. So this one is the only super resolution microscope in Algeria. And it also has the option to do live cell imaging. Then we also have this specific microscope, which is normal inverse the fluorescence, epifluorescence microscope, but it has a unit for photo patterning. So photo patterning is to create different patterns to try to recreate the microenvironments of the cells. So here you can see, so this is sorry, it doesn't work. It's supposed to be a video, but we can control where the cells go on the micropatterns. So with this device, we can pattern and image at the same time. And this one was the photo patterning unit was the invention of Professor Azion. And it's unique in Africa. So as you can see here in the map, it's the only one in Africa. It's also hosted in top-notch universities like MIT and Berkeley, etc. Then we also have super resolution scanners because we work on micropatterning and cells microarrays, well, cells, proteins, or DNA microarrays. So we do the fabrication, we also do the development of these systems. And this one is a large-scale scanner for microarrays. Now, as for the research that we carry at the platform, so we work on genetic, cellular, and tissue engineering, and we try to include all of these on chips. So these that you see here are microfluidics chips that can be used for different applications in health, precision medicine specifically, or diagnostics, agro-food, environment, and industrial applications. Because here we miniaturize the reactions, and you can do large-scale experiments on a chip. We also work on cells micropatterning. So we control the microenvironment either on 2D or 3D level. And this one here, you can see cells on different micropatterns. And the aim is really to mimic the natural microenvironment. And when we mimic the natural microenvironment, then when we do drug testing, for example, we will get more specific response from the cells. And we also do double patterning. So this is going to be a patented technique, because right now the challenge is to put different types of cells on the same chip, and to do it and manufacture it at industrial level. And you can do different types of cells with Primo, the photo patterning microscope that I showed you earlier, but you can only do it on a spot. So we found a solution to do this on large scale. We also work on 3D bioprinting. So we have 3D bioprinters at the Bioengineering platform, where we can print different types of cells. And we're working on developing hydrogels that can let you culture different types of cells, and to get the spheroid models, which are the nowadays models for drug testing and different applications. We also developed a technique to do spheroids microarrays. Okay, so for the pros of our platform, so the main thing is that it's the platform at the Biotechnology Research Center is hosted with other platforms as well. So we also have a genomics platform with top-notch equipment. So we will soon receive the Illumina Next Gen X. We also have proteomics, a very developed and fully operational proteomics unit. So you have all these technologies in one spot. What really makes you able to streamline all the process of the work. We also have the Biotechnology Research Center has different divisions, so it's also cross-sectorial. So we work on health, agriculture, agri-food, environment, and industrial applications. And we also are developing a biotechnology hub at Constantine, where we have the National Higher School of Biotechnology, of which I'm a graduate. And we also have the Biotechnology Research Center. So we're trying to do the teaching with research. So we always host students workshops at the Biotechnology Research Center. And even that, the platforms are really dedicated for big projects, but we train our students on the different equipments, and we give them access to really work on giving them insights on what they can do in the future, in their future research. So we also have active collaborations, specifically for bioimaging. We have collaborations with the International Genetic Engineering and Biotechnology Center. We have an ongoing collaboration with the University of Southampton to develop teaching program, because we believe that capacity building starts with teaching programs. And then we also have the I4All grant recently. I will be visiting Dr. Samira's lab, the MHM ENS Paris platform, bioimaging, global bioimaging grant. Okay, so what we're really looking forward to right now is to upgrade the infrastructure, because we do not have a confocal microscopy in Algeria. However, we're working on 3D cultures, and we do really need to upgrade to confocal and light sheet imaging systems. We also looking forward to strengthening the bioimaging data analysis, and we had recently the fund to start an AI platform. The AI platform is going to be next to the bioengineering platform, and also proteomics and genomics platforms. Work on integrating all this data and working on predictive models. Also looking forward to strengthening the training programs, as I've mentioned, with the collaboration with Southampton University. So what we would like really to do right now is to do this with different African universities to develop shared training programs, and also contribute to the global bioimaging shared infrastructure. Okay, so thank you for your attention. Here, this is the Biotechnology Research Center, the National High School of Biotechnology, and this is Mimical. This is to show you the biotech hub we are working on. So research, we developed and we created our first Biotech 4.0 startup, Mimical. So thank you for your attention.
Thank you so much, Wiame. We're going to spare the questions after Georgina Fletcher's talk.